Hi, I'm D.Simberloff from the University of Tennessee. I wish I could have been there in Paris to deliver this talk in person, en français, but something got in the way, and I will do my best to do this via Zoom. And I apologize for my American English. I'm going to talk about a prediction that was made by the American nature writer David Quammen in 1998. He predicted that in about 150 years, the Earth would still be green, but it would be the same species everywhere because of biological invasions. It would be what he called the global weeds, plants and weedy animals as well. Around the same time, the ecologist Jack Putz popularized the term homogecene to characterize what he thought was happening with the spread of invasive species homogenizing the Earth. And that became a growing part of the concern with mass extinction, and especially the role of invasions in global biodiversity loss. And what I'm going to do is to assess these predictions of ultimately homogenization and a planet of weeds. The problem is that both metaphors conjure up an image of an endless landscape dominated by non-native plants, same species common over large swaths of the earth. But despite a very large and rapidly growing literature predicting the trajectory of various invasions, the predictions don't really address this planet of weeds image. Instead, they focus largely on the future spread of particular species or the number of native species that will be found in particular areas, and not on the coverage by these species. So, for example, this large summary by Chitri and his co-authors predicting the levels of invasion by plants in Europe in 2020, 2050, and 2080, as you can see, shows invasions increasing, but the levels of invasion are percentages of vascular plant species that are aliens. And the increases and decreases are presented as positive or negative percentage change in the levels of invasion over 2020 in terms of percentages of the plant species that are aliens. Well, most scenarios predict with various kinds of land cover change that levels of invasion by this measure will increase for most of Europe, and one could reasonably hypothesize that a greater percentage of the flora consisting of non-native species would correlate with greater aerial coverage, but that would just be a hypothesis. It would have to be tested. Similarly, this review by Lambden and co-authors of the temporal trends in alien flora of Europe, showing the coverage of plant data is in terms of the number of species recorded as alien in at least one European nation. So it's in terms of number of alien species. And the map, as you can see, is a map of geographical coverage of plant data, not geographical coverage by plants. Similarly, for North America, this paper by Beth Bradley and her co-authors, it looks at all the invasive plants. It looks at all the alien plants, of which a subset are termed invasive, and compared them to native plants. And what you see here is for each one, they predicted by environmental niche models the potential range, and then the fraction of the potential range that's actually occupied. And so you can see that the invasive plants, first of all, They tend to have larger potential ranges. And secondly, the ranges are less full, the potential ranges. They're not occupied yet. So the idea is clearly they're going to spread. And so again, one could reasonably hypothesize that if invasive plants are going to, or alien plants are going to be spreading and covering and are living in, occupying larger fractions of the United States, that the mean cover by alien plants would be increasing. But that's just a hypothesis. Again, it's not about cover. It's about range. The type of data that would be needed to assess the Planet of Weeds hypothesis would be mean cover by these various species. Well, mean cover data are typically collected by ecologists working at very small scales, but they're not gathered regionally. It's possible that with the continuing development of remote sensing technology, and use of drones, such data might become accessible over vast areas. But we don't have data on mean cover now. We just have these range data, and the range data don't tell us about coverage. Certainly, the numbers of invasions has accelerated over the past several decades, and there's every reason to think it's going to continue to do so for three main reasons. The most important one is habitat destruction and transformation. And here you can see between the years 1800 and 2000 in this paper by Hanno Siebens et al. That accumulation of alien species going up worldwide. And here's for the different continents and for different groups: mollusks, insects, fishes, birds. In general, the lines are going up, and they don't show any saturation. And largely, the single greatest reason is habitat destruction and transformation. The second reason they're going up, and will continue to do so, is increased travel and trade, and its role in both deliberate and inadvertent invasions, and we can't really doubt it. For example, here are the data for containers carried by ships. TEU is the 20-foot equivalent unit. That's the standard container that you see on ships. And as you can see, the prediction is here you see 1996, and it went up roughly fourfold to 2016, and the prediction is that it's going to go up somewhere between two or threefold and about fivefold. Well, of course, in the past, during the global recession of 2007 through 2009, the container growth slowed down, and obviously, with the COVID-19 pandemic, there's also going to be a decrease. But as you can see here, there was a recovery fairly quickly, and in container amount of containers being shipped, and that's probably what will happen here also. They're very similar statistics for also for air transport, of course, but these containers are the biggest amount of cargo being shipped. The third reason that this that the spread of invasive species is clearly going to grow is climate change, and there have been many attempts to show for particular species or groups of species that climate change is going to increase their likelihood of invading and their range in the invaded area. So one of the key papers is this one by, again, by Bethany Bradley and her co-authors in 2009, I believe it was. And here's one example. She's looking at cheatgrass, probably the single worst invasive plant in North America. The upper map shows the potential climate as assessed by environmental niche modeling, and the actual actual range of cheatgrass is in black. And then the second map, the map below it, shows the future invasion risk as climate changes by doing, again, environmental niche modeling exercise. And you can see the range is likely to expand, and the risk of invasion over the range, the potential range, is quite great. And there are many papers. This was one of the first ones, but over the last decade, innumerable studies predicting the spread of particular invasive species with climate change. So the sum of the evidence from these three factors, namely habitat destruction and transformation, trade and travel, and climate change, really make it clear there's going to be an overall increase in the number of invaders and the range, the geographic range, over at least the next century. Well, does that mean a future planet of weeds, as was predicted within about 150 years by David Quammen? Well, there's some other things to consider. There's this issue of biological inertia. This idea was first proposed in a review by Pearsall of Elton's ecology book was published in 1958. And Pearsall said that, yes, of course, the number of introduced species is increasing and they're going to spread, but he didn't think that this would happen very quickly, because he felt that native vegetation, especially dense native vegetation, would largely restrict the spread of invasive plants anyway to places where disturbance removed the natives. And he suggested for forests it might take even 500 years, 5 centuries, before there would really be appreciable change in climax forests. Then a paper by Betsy Van Holly and Hazel Delcourt and me in 2003 also raised this issue of biological inertia, especially for forests, because of the longevity of many forest trees that live hundreds of years, many hundreds of years in some cases, and also the legacy they leave of their presence in chemicals and various soil properties, etc., so that even an aggressive invader might need centuries in order to replace begin to invade an area. And there are many Holocene palynological studies that depict centuries or even millennia-long slog for a very climate-induced range expansion of some tree species to spread to where it's going to spread because of the climate change, probably requiring disturbances like tree falls or fires to remove some of the native species. Also, the paper by Downey and Richardson, gives many examples of soil seed banks that can last not only decades but centuries, and again will provide some sort of inertia or it tend to impede the spread of invasive species, even if ultimately they are going to spread. This paper by Early et al., sketching out the threats of invasive species over the next century, I think also supports this idea. They looked at the threat of introduction, threat of establishment, et cetera, but the key figure is A, the one at the top. And although there aren't many parts of the Earth that have very low, VL stands for very low threats, you can see very large parts of the Earth have low, the light blue threats of invasion in their view from their various analyses over the next century. Another point to bear in mind on a personal level, and I suspect many of you have had similar experiences, and that is if you go hiking in a large forested natural area and you see many, many invasive non-native plant species along trails and in other disturbed areas. And so I often go hiking in the Great Smoky Mountains National Park, which is not far from the University of Tennessee, where I am. It's this huge, huge national park. It's the size of a small nation, really. And there are lots of non-native plant species there, somewhere around 380. But in this picture, you won't see any of them. And if you walk along a roadside or a well-used trail or right around nature centers, you see lots of, you can easily see 10 or 15 non-native plant species, some of them quite frequent, almost dominant. But if you go into a roadless area just a few meters away, 10, 15 meters away, you see very few. And if you go 50 meters away, you see none. You'd have to look for a long time to find a non-native plant there. And most of the intact forest in this national park is uninvaded. Now, how long it will take, or how long the inertia will impede invasion in the face of the forces that I talked about a moment ago, is unknown. But I doubt that we're going to see a planet of weeds in this century, or even in 150 years. And I suspect this forest will look pretty much like this, at least for a century from now. However, I suspect that without new policies and new management approaches and willingness to implement a global strategy to deal with invasions, eventually 5 or 10 centuries from now, this will look very different and Quammen's metaphor will have been realized. However, there are many existing technologies that could greatly impede the achievement of a planet of weeds, maybe even stop it in its tracks. And there are new technologies on the horizon that also might help. I don't have time to talk about all the existing technologies that and the improvements, the incremental improvements of all of them. The three main ones, of course, are various forms of mechanical or physical control, the use of chemicals, and then biological control. And then there are various more specialized approaches that are used on particular groups of organisms, like pheromones, hormones interfering with mating, sterile male technique, etc. And for all of these, there have been a series of incremental improvements that make them more effective, that often decrease the likelihood of non-target impacts and increasing the likelihood that these really will have the effects that we hope. But there are, and maybe they would help to turn this, what looks like a losing battle against invasions, into at least a persistent stalemate. In particular, there are lots of proposed new technologies, some of them already underway, many of them involving molecular genetics, that I think could be game changers and could greatly improve our possibilities of preventing the planet of weeds. So, I mean, a very simple one is this problem of early detection, rapid response, which is very important if we're going to stop invasions. We can do it much more readily if we find them early, but it's often hard to find them early because there just aren't enough paid personnel to do this. But the advent of smartphones and various apps like iNaturalist, and in the US, I've Got One, which you can put on any iPhone or Android phone, and citizen scientists or even citizen non-scientists can go and if they see something odd, photograph it. And it's happening a lot in some areas, including in the US. Of course, there has to be some stable program to receive, to register, and to evaluate the images. In the US, it's EDMAPS, which accepts all of these reports and has a procedure for analyzing them. Another new technology that is greatly expanding the range, certainly of detection, is the various use of drones, and not only drones. There's the idea that mini satellites can begin to do this and various other kinds of aircraft, but drones are already being used to monitor for new invasions, to monitor the spread of invasions. In some cases, actually to aid management after the invasions are discovered. A really important new technology is the advent of eDNA, environmental DNA. It's been used largely to detect invasive aquatic animals, and it's really revolutionized the detection of invasive aquatic animals. And although plants and terrestrial species in general have presented challenges, there are several research teams that have more recently used eDNA to detect invasive aquatic plants and some terrestrial plants and insects, for that matter, on terrestrial plants. And notice the picture in the lower right. I have an arrow to it. It's a very recent study that used pollen pellets from honeybee hive to detect invasive plants. So there are many possibilities to increase the range of organisms that can be detected with eDNA. And of course, there are entire journals of using eDNA and how to use eDNA. Another approach that's already now underway and got underway very quickly is gene silencing. And of course, there was very substantial interest in gene silencing for medical purposes and to some extent agriculture. And its use in dealing with invasions is sort of piggybacked on advances, especially in medicine. The original idea is captured in this paper that was pretty recently published, 2016, was to have the gene silencing entity, usually double-stranded RNA, in some substance that could be eaten by the targeted invasive species, usually an insect, but it was also has been worked on for rodents, for example, and to get it in a form that enough of it will pass through the digestive tract into the bloodstream, it will actually kill the target. And there was a lot of progress made on this. This one particular paper is talking about the Colorado potato beetle and how this is really working. But actually, that's sort of an old-fashioned approach now, as a number of teams have been combining gene silencing with the use of transgenes in order to get a transgene that into a crop plant, such that when the targeted invasive species, in this case it's the corn rootworm, bites into the plant, the transgene is activated and produces the gene silencing agent, which kills the plant. And Monsanto was probably the first off the block actually to produce such a thing in maize. And here is a report of how it was approved by the Environmental Protection Agency of the United States. But Monsanto, this is just one case. DuPont filed a patent application last year to do gene silencing in exactly the same way to attack pentatomids, stink bugs, including the brown marmorated stink bug, which is a major invader in eastern North America now. And again, it's the idea of introducing a transgene into a host plant, such that when the stink bug attacks the host plant, the the transgene produces a silencing agent and kills the invader. And maybe the most remarkable use of this to date was just reported 3 months ago in Science. What this team did was they modified the genome of a bacterium that's in honeybee guts. It's a gut symbiont of honeybees, and they modified that bacterium with a transgene to express double-stranded RNA sequences for genes of the varroa mite, the parasitic varroa mite. That's a major threat to honeybees. And so it kills the mite by activating the RNA interference mechanism, because the mite ingests the bacterium which carries the gene silencing sequence for the mite, and so the mite dies. This has so far been used on animals, especially insects, and animals are targeted to use this mechanism. The only plant I know that's been developed, or where they're trying to develop a method like involving gene silencing, is Phragmites australis, which is a major invader in North America. Again, using double-stranded RNA, and not on transcription as here, but in translation. And then, transgenes generally, even aside from gene silencing, can be used in many ways. I guess the whole idea of using genetically modified species with transgenes burst into public view around 10 years ago with the famous Oxitec friendly mosquito, which is Aedes aegypti, the vector of dengue and of yellow fever and chikungunya. And what Oxitec did was they introduced a transgene that's inactivated by tetracycline, but it renders the females flightless, which would make them lethal in nature if the diet doesn't have tetracycline. So they rear huge numbers of them, sort them out, throw out the females, release the males into nature, and the males mate with the females, but all of the offspring of the wild females then have that gene, and they die. And reports of the first field test of this construct, this friendly mosquito, and Grand Cayman, raised a lot of concern about lack of regulation, possible unintended consequences. But the advent of Zika led to massive release of these in Brazil and especially in Brazil, and it worked. Great decline in the number of Aedes aegypti and in areas where these were released. Now, of course, the recent discovery that some of the genes from these actually are now in wild populations of Aedes aegypti in parts of Brazil, not the transgene itself, but genes from the transgenic strain, have led to a further controversy. And Oxitec has responded saying this is no big deal. But the point is, they produced a construct, and it actually did work to knock back the Aedes aegypti population. And Oxitec is moving ahead. They are producing very similar constructs for several other insects, including all of these very invasive species, the medfly, the spotted winged fly, etc., etc. And there are transgene projects being mounted against other invasive animals like the Pacific oyster, the channel catfish, the common carp, in various stages of development. The only case I know of in which an invasive plant, is being targeted by this means is the idea to use it against Scotch broom in the United States. It's sort of odd because transgenes have been used in plants a lot in order to introduce traits that are useful, like pest resistance, herbicide resistance, drought tolerance, salt tolerance, increased yields, etc., etc. But they don't seem to have been used to try to control invasive plants the way they are being used now for animals. And even more striking is some of the plants that have been in which transgenes have been used to introduce properties are very invasive, but the properties they've introduced were not to stop the invasion. So, for example, Phragmites australis, they introduced transgene to reduce lignin content to facilitate its use as a biofuel feedstock, or Spartina alterniflora to improve mercury phytoremediation performance, not to attempt to control its invasion, except for the case of Scotch broom. And now, of course, with the advent of gene editing and CRISPR, there are, a much greater range of possibilities, and this was recognized quite early as CRISPR burst on the scene in this particular paper by Esvelt et al., which I believe was 2016, pointing to several ways in which CRISPR-Cas9 and also other RNA-guided gene drives could aid here. And CRISPR now is there are entire issues of science about it. Again, it's the invasive species community is sort of piggybacking on great advances in the medical field and also in agriculture. And again, there's a whole journal now for CRISPR. So this is exploding on the scene, and there are improvements every day. There are at least two very well-funded projects using CRISPR, Cas9 gene editing, to control invasives. One is this G-Bird project to control invasive rodents, beginning with house mice, but ultimately aiming also at invasive rats and other rodents. And the other is target malaria, using CRISPR-Cas9 gene editing to target Anopheles mosquitoes that transmit malaria. And target malaria has made a lot of progress. It's very well funded, and so this is going to happen. A very striking recent project that's funded to a small extent by the U.S. federal government is using CRISPR-Cas9 to try to eradicate or at least greatly knock back quagga and zebra mussels by introducing a transmissible cancer of all things. The transmissible cancer is known in certain soft-shell clams, and it can be carried into zebra mussels and quagga mussels. And the CRISPR-Cas9 is being used to suppress a tumor silencing gene in the mussels. I don't know how far this will go, but they are actually trying to develop this at a university in Ohio. And of course, transmissible cancer is the very same kind of disease that's greatly threatening the Tasmanian devil. And so we're talking about the same sort of thing. Well, this whole approach, obviously, and the obvious possibility of some unintended consequence and non-target impact has led this entire idea of gene editing and the use of transgenes to be quite controversial, including the last World Conservation Congress, where Jane Goodall and others circulated a manifesto signed by a number of people saying we should never use this for a conservation purpose, which would even include dealing with invasive species. And of course, there's the possibility that, as gene editing becomes easier and easier to do, and we get kits, that we can order online, and, people eventually will be able to do it in their garages, I guess, that there could be an accident or someone could use it for an eco-terrorism goal. And so there's a lot of worry about this. The U.S. National Academy Research Arm, the National Research Council, did a study saying, yep, it's something to worry about, but the possibilities of good results are sufficient that we should continue with laboratory research and field trials, even though we're concerned. Similar UN treaty doesn't really stop it. It just sort of expresses concern. So, it's happening even though we're worried about it. So I'll sum up about what does all this portend for a planet of weeds? Will the Earth really ultimately be homogenized? And if so, when is this going to happen? Surely more and more of the Earth is going to have more and more invasive non-native plant species and some animal species, and some fraction will be dominated. But the Earth will still be green, I think, as Quammen said it would. How much will be global weeds? Well, I don't know. I think we could greatly impede this, the realization of this metaphor, if there were a global strategy that we could all, where nations would collaborate and cooperate, and the various management tools in place or on the horizon were implemented. So I think this could happen, but it's difficult for me to be optimistic about this, especially in an era of growing nationalism. And, the response to global climate change also does not inspire great confidence about a global strategy actually being implemented. I think some of the various new genetic approaches currently being researched and other technologies we haven't even dreamed of will be employed to some extent. So I think certainly in the 150 years that Quammen was predicting, I still think that much of the Earth will be dominated by native species. But I think the forces arrayed against native biota are so massive and inexorable that it's hard to be optimistic that we won't have a planet of weeds in 500 years or a millennium. So that's my story, and I'm sorry I can't take questions, but my email is available if we want to discuss this.